Okay, well, first of all, thank you for coming to our event. We're really happy that you've come to listen to us talk. We're going to be, like the title says, talking about how we use health data to understand heart disease and COVID-19 risk. In just a minute, I'll introduce our speakers, but first just a little bit of housekeeping. We are recording this session, but we can't see or hear the audience, so we aren't recording you, just us. And we do have a Q&A function. So if you have any questions, that's going to be the best place to put your questions. We'll try and answer them as we go, but we'll also have a Q&A session at the end where we'll be answering things verbally rather than in text. But that'll still be the best place to put the questions, and we'll hopefully come back to them at the end. There are three of us who are going to be speaking. So first of all, we've got me, Amy. I'm now, all of us work for the cardiovascular epidemiology unit. That fundamentally is a lot of long words that mean we're interested in brains and hearts and how they are affected by things on a sort of population level of risk rather than individual level. I'm a statistician, and I'm going to be talking to you briefly, just in sort of introductory sense, about what is health data. Then we have Samantha, who's a research associate with the unit, and she's going to be talking about the CVD COVID UK study for about 15 minutes. And then finally, our other Sam, who researches around polygenic risk scores, is going to talk about 20 minutes on genetic risk and how it can be used to understand our likely future health outcomes. And after that, we will be answering your questions. As I say, best place to put those is in the Q&A. If you have any problems around video or hearing or what's going on, feel free also to put stuff in chat. But in terms of questions that are related to the talk, better to go in the Q&A. Okay, so what is health data? Probably you already have some idea because you have been, decided to come to this talk. The obvious answer is it's data about people's health. Right, I'm done, on to the next. Oh, that's not enough? You want to know more? Well, health data can be a lot of different things. It could be patient data from the NHS or social care with information like patterns of admissions to hospital or what different drugs different GP surgeries prescribe. A lot of data is kept in our health records, which makes the UK patient records like this really rich source of health data. It can come from research studies, so studies on groups of people with a particular disease or who are testing out a new drug. It can also come from collections of blood and tissue samples. So this might have been collected as part of one of those research studies, or it could have been collected through a private company like 23andMe, who has a gigantic collection of genetic data, and it's not super creepy. Maybe it's super creepy. Similarly, there's a lot of data in medical images as well, so things like MRI scans or CT scans. Also, increasingly, we're using apps and devices to monitor our health on our phones. And through those, it's possible to build up incredibly detailed data sets of heart rates and blood pressure at the time. My partner has an extensive collection of exactly what their heart rate does during weekly runs, matched up to speed and elevation and whether they're leaning more to the left or the right. Similarly, I met a girl recently who real-time monitors her glucose levels because she's diabetic, and she can share that data with her family so that they know she's okay all day. And finally, there's another category, which I've just literally called other. And this is the sort of data where we don't necessarily think of it as health data, but actually it can be really useful. So one of the weirder ones I know about is you can actually track, to some extent, the rates of COVID by the rating of Glade candles on Amazon. Because when the rates of COVID go up really high, you get a vast number of complaints that the candles don't smell, tracking really nicely with the actual rates in the population. So it's sort of amazing how much data of the data we share with Google and Facebook and so forth can actually be used to unpick the mysteries of health at a population level. Now, obviously, health data is both sensitive and personal. Members of the public have a lot of concerns about how it might be used or misused. and people really want to know that their health data is kept safe and only used in ways that benefit the public. And to do that, we ensure that all our data is collected and used ethically. For me, the most important principles around health data are obviously consent, ideally that everyone has agreed to this data being used in this way, anonymity, that we can't identify particular people from our data set. We're only thinking about the impact on the population, or a large number of people. We're not looking at a specific person and knowing things about them. And then security, the idea that you only want the right people to be accessing the data. So you only want the approved researchers accessing it and the data not to go anywhere else. And finally, an important one for me is utility. It needs to be used appropriately and where it's going to benefit other people. The majority of the data sets we use in our department are essentially opt-in studies that have been created or that we have got permission to use, where they've been gathered from people who have explicitly consented to join, and then for us to use their genetic data and to ask the NHS for their hospital data or GP records. They can withdraw their data at any time and we would stop using it. Working with health data, particularly health data that comes from multiple sources, can be surprisingly tricky. The data might not be complete, so you might have blood results in some people than other people. It might have contradictory statements. I've got a data set where, because people have been surveyed multiple times, with one person who said they got diabetes when they were 50, unfortunate but believable, and then 2 years later said they got diabetes when they were 8. And you sort of look at that and think, well, they can't have forgot an entire lifetime of diabetes the first time they were asked. So, but which one of these is true? And you don't have necessarily any more information to understand that. But what we have to do is try and use the clues in the data to make our best guess at the truth. Okay, so if it's sometimes missing and messy and isn't necessarily exactly the thing we want, why do we use health data? Well, health data helps us understand disease and conditions, what causes them, how many people have them, how to progress. And it can help us find new ways of treating them or spotting them earlier. There are so many examples of how health data science has improved our lives. One of my favorite examples, they used machine learning technology on thousands of eye scans, and it used it to, now they can use it to identify signs of eye disease earlier than people, other than doctors looking at the eyes would be able to spot it. And it helps recommend which patients should be referred on for more care or what care they should be referred on to. Similarly, Sam will discuss in a minute how patient data around vaccines help show that COVID vaccines are both safe and usually reduce the risk of severe COVID symptoms. So health data can be incredibly powerful, and that's why we need to use it. I'm just going to quickly talk about a couple of the data sets we use in the department I am familiar with. So one is interval, and I am only going to talk about this very quickly because I don't have much time left. Fundamentally, there's about 50,000 blood donors who were recruited between 2012 and 2014. It contains information like what their lifestyle is like, we've got their genetic information, we've got blood results. Another data set we have, which I love so much, is UK Biobank. It's made up of about 500,000 volunteers. Unfortunately, they're all aged between about 40 and 69, so there's a bunch of data we don't get because we don't really have any children or teenagers in there. But what we do get is some really incredibly rich data. So it's got MRI scans, not MRI scans where they've been collected from hospital data, but where they've taken everyone and sent them to get an MRI scan whether they needed it or not, so that we have the data. But it's also got data like, how many avocados did you eat yesterday? And it's that sort of beautiful combination of it's got vast amounts of medical data, but also really, really precisely answered slightly odd questions that makes it really fascinating to work with. I said that most of the data sets we used we collected from people consented from them. There are also other data sets that are used in the UK. They're used in what's called trusted research environments. And in particular around COVID, there's one that they just took everyone's records and put them in because it was so important that we try and get answers out about COVID quickly. And Sam will be talking about this a bit later. And the idea of trusted research environments is it's a secure computing environment. Traditionally, researchers download data sets, we use it, do our analysis, we can see the whole thing. But in the trusted research environments, the data stays in the location and we access it remotely. And the idea here is there's a bunch of principles that make this safe, that only accredited trained researchers use it, that it's only used for ethical purposes, that it can only be accessed through the secure system, that it never leaves the TRA, and that the data is all anonymized so that none of the researchers can see any particular person's data. And importantly, their access could be revoked at any time if they don't behave appropriately. So what do we use those sorts of environments for? Well, for that, I'm going to hand over to Samantha, who's going to talk a bit about the CVD COVID UK research study. You need to unmute, Sam.
Hi, yeah, thanks, Amy. So this is, so hello, I'm one of the Sams today, and I'm going to talk about, I'm a research associate at the Cardiovascular Epidemiology Unit, as well as the Center for Cancer Genetic Epidemiology. And today I'm going to briefly introduce you to two pretty fun lines of research at the Cardiovascular Unit. So cardiovascular...
When you need to swap screens.
Oh, sorry.
No worries.
How does that?
Beside the X. No.
Okay, let me try again.
Perfect.
Okay, brilliant.
Perfect.
Sorry. So yes, yeah, we were here. Yes, so I'd like to briefly introduce you to two pretty fun lines of research here at the unit. The first topic will be cardiovascular disease in the context of COVID, as Amy mentioned, where we'll answer the hottest COVID questions, be it to do with infection or vaccines, using the near real-time UK-wide data. And the second topic will be on machine learning, which is all the rage in every field pretty much, but in the context of healthcare. So since cardiovascular disease is a whole mouthful, and I struggle to say it, so I'm going to call it CVD from now on. So let's start with the COVID and the near real-time population-wide data studies. So you may have heard of the COVID pandemic. Well, it's a major global challenge with long-term impacts on population health, healthcare services, and the wider economy, as you might expect. So what we want to do is to understand the impacts of the pandemic on cardiovascular diseases. So there are some direct impacts, for example, directly to do with COVID infection or potentially its vaccinations, and we'll explore this a bit more here. Or indirect stuff, for example, how the government and the health services respond to the pandemic will have impacts on, for example, how the CVD presents, their diagnoses, how the management and outcomes of the diseases. So, for example, you can think you might expect a dramatic drop in the number of people going to hospital with heart attack and stroke, or patients might be arriving too late for acute treatments that could otherwise probably really help them and prevent further complications, for example, clot removal and stuff like that. So what we want to do is to enable the government and the NHS to make evidence-based policy decisions to mitigate these problems as much as possible, and also to inform clinical care. So here's some questions that we could ask amongst many about in the context of COVID. So COVID infection is known clinically to encourage blood clots and inflammation. So we can ask, how does getting COVID affect our short, medium, and long-term risks of serious cardiovascular diseases? Or you may have heard of some reported cases of blood clots at odd sites, such as the cerebral veins, following COVID vaccination. So what does population level analyses tell us about the risks of these rare complications after vaccination? And should we really be that worried about it? And also there are floods of papers coming out during the pandemic asking very similar questions. and we see a wide variety of approaches being used, and their conclusions don't necessarily always fully agree. So one of the studies we're doing is to implement these models on the same population-wide data using consistent definitions and everything, and compare the results of the different methods to see whether they agree and to what extent do they agree. And if they diverge, why do they diverge? Are there theoretical assumptions not met, etc.? And how would that affect our interpretation of the results and what we should do? So in order to study all these things properly, we really need to analyze up-to-date representative population-wide, population-level data, which will give us a representative evolving picture of what's happening. And this big data gives us the statistical power to study the rare outcomes, like the clotting at the rare sites, which normally happens on the order of 1 in a million, which is a really tiny number, and you need a lot of people to study that properly. So we're lucky to have the CVD COVID UK Consortium, which is a research program that wants to study the impacts of COVID, infection, medications, vaccines, etc., on CVD and vice versa. So they worked with the national data providers such as NHS Digital England to get some nationally collated data across the 4 nations of the UK. And they bring together data sets that are usually kept separate. For example, GP data, hospital data, COVID lab tests and vaccinations, deaths, medications, ICU data and stuff like that. So I mainly work with the English data, and that covers a population of around 57 million people in England, from over 98% of English GPs. The consortium really values transparency, so all analyses, plans and reports are publicly available via the HDR UK website. And our findings are reported to SAGE, which is the Scientific Advisory Group for Emergencies, NICE, the National Institute of Health and Care Excellence, the Medicines and Healthcare Products Regulatory Industry Agency, and others. So what did we find so far? So here are some very high-level kind of results. As the doctors expected, COVID infection is associated with an increased risk in a wide variety of clotting diseases, some with rather long-term effects. And you may have heard of long COVID, in fact. And here I show 2 umbrella outcomes. So the arterial stuff is on the left, which has to do with blood vessels bringing blood away from the heart, and venous on the right, where you have blood vessels bringing blood to the heart. And you see you're 4 times more likely, especially in the early times, right after getting COVID, to get for you to get arterial diseases. And yeah, so what about vaccine complications? So most clotting diseases, we actually see that they maintain the same or even lowered risks after vaccination. While we see an increase in what you call relative risk in 2 rare types of clotting problems, which is the clotting in cerebral vein, which is the thing that is mostly in the news. And then you also see some lowered blood platelet counts after AstraZeneca vaccination for the under 70s. But it's important to remember that the outcomes are normally very rare. So this relative risk is really comparing 2 very tiny numbers. So the increase in relative risk translates to a tiny excess absolute risk. So clotting in the cerebral vein is of the order of 1 in a million within the first 4 weeks post-vaccination. And the lowered platelet count is around 6 in a million kind of thing. So to give you an idea of the unit of 1 in a million, that is really comparable to your chance of getting 20 heads in a row of flipping a coin. So assuming that you are more honest than Two-Face, I'm doing a Batman theme here. And according to the Berkeley Stats Department, other 1 in a million chance things include the chance that the next 1 in the next 24 babies born in the US will become the US president, or the chance of you dying due to a ski and snowboarding accident during a one-day visit to an official ski area. So that's pretty rare, as you see. And these risks are really dwarfed by the very real risk of getting COVID with its consequences and very known consequences, dying from COVID or inflicting COVID on other people by spreading it. So I've described some big data and so showed some things that are what it can tell us using statistical methods. So let's turn to another exciting potential that this kind of big rich data can give us, which is to use it in machine learning in healthcare. So I'm sure buzzwords like machine learning, artificial intelligence are omnipresent, and unsurprisingly, it's also a big thing in medicine these days, with a potential to do some good for the world, hopefully. So I'm not going to get into the whole what's intelligence thing because it's an ongoing debate, but artificial intelligence, AI, has to do with basically machines taking in information from the environments and taking the appropriate actions to maximize their chances of achieving some goals. This is a definition we're going to run by for our purposes here. And machine learning, ML, is a branch of AI that allows machines to learn from the data without you explicitly programming its responses, for example, what it should learn. So why use machine learning at all? Don't we have lots of traditional tools? So currently some methods that are used to make referrals include checklists. So GPs look down a checklist and see whether or not how much you've satisfied this checklist and decide whether or not to give you a referral. We also have statistical models that can flag some high-risk people, for example. But these methods can get quite overwhelmed, especially if you want to build very high-dimensional models, for example, using images, or even when you want to include many potential predictors from lots of different sources of very different natures of data, etc., for risk prediction. So ML can also help with making more targeted predictions and recommendations. So it can do the high dimensional thing, which means it can consider a rich set of individual specific characteristics. And this is what we call precision medicine. Machine learning is also flexible and can find quite complex and model complex hidden patterns, trends, find subtypes of diseases, for example, by how they progress, what groups of symptoms tend to happen together, and things like that. And we might not actually know the answer here. So ML can help us form, in fact, some kind of data-driven hypotheses, which can be quite interesting for future research. And this, and using ML can let us really make use of the abundant raw electronic health data just lying around and hopefully use it for some good. So how can this work in the real world? Machine learning tools can be helpful sidekicks to clinicians and healthcare providers. So to start with, you might have clinicians or healthcare providers or policymakers having some questions. For example, they might want to split the population into high versus low risk groups, or for more efficient interventions. So you can invite the high risk groups in for more invasive and expensive tests and leave the low risk group alone. So then what we would do is we want to then use the already collected electronic health data records, including, for example, people's health history, diagnoses, test results, the age, sex, and so on. And then we would develop a model, which in a process which clinicians should really be a very integral part of. And we will work with them to formulate very specific questions, design the models, and see what should go into the models. And we can then train the model, which can potentially do various tasks. For example, it could predict risk or even trajectories of risk over time. It could maybe identify subgroups of diseases by their trajectories, other diseases associated with it, symptoms, and so on. It might be able to recommend diagnoses, considering a comprehensive list of options. It could predict a patient's responses to various treatment courses, and therefore help doctors make the best treatment decision for a particular patient. So since medicine is quite high stakes, we want a few more things apart from just high predictive accuracy. So we also want it to be interpretable so that clinicians can understand why the model made a certain prediction or recommendation, to make sure the model didn't pick up some false associations or biases from the data. And we want to, and knowing why a certain prediction or recommendation is made might also help with treatment decisions. We also want it to be fair, so we don't want it to discriminate against certain disadvantaged minority groups, for example. And this really goes beyond the idea of equality, where everyone is treated the same, but rather we want to really strive for equity, where we take into account more individual specific needs to get so that everyone has the same chance to achieve their health potential, if that makes any sense. So if all of these go well and we have a very good, well-tested, robust model, this could have a real impact on the world, where we could potentially empower clinicians and healthcare providers. We might be able to save and improve lives while saving the NHS money by being way more efficient at resource allocation and intervene more effectively. And we might even come up with some data-driven hypotheses to drive future research, and so that we can understand various diseases much better. So here are a few collaborators here at Cambridge involved in these projects I mentioned. Of course, there's many other amazing people to thank, especially collaborators from the CVD COVID UK consortium that I mentioned earlier. So thanks for listening, and I'm going to pass the baton on to Sam Lambert, who's going to talk about genetic stuff.
Great. Thanks, Samantha, for your presentation. Yeah, it was really interesting about COVID. And yeah, I'll get started with sharing my screen. Am I sharing the right screen? Amy or Sam, could you give me a thumbs up? Oh, great. So yeah, hi everyone. I'm Sam Lambert. I'm a senior research associate in the cardiovascular epidemiology unit. And today I'm going to be talking about predicting and understanding disease risk using genetics and how we use health data to study that. So I think, people might appreciate that in the population, there's definitely traits like body mass index, so how heavy you are, height, blood pressure, or disease risk that vary among individuals. And these traits and disease risks sort of vary in different ways. We know that certain diseases are more common to people who are older, certain diseases are common more in men versus women. And these diseases sometimes are associated with lifestyle factors. So how much you exercise, your diet, smoking, or your environment. So air quality could set off asthma and things like that. And these risks or these variations in traits and whether you're likely to get them to some extent rely on your genetics or the DNA you are born with. So there's lots of different types of genetic variation, but it's more useful to think that, the DNA, we each have 22 autosomes and 2 sex chromosomes, and any two individuals have 99.5% of their DNA identical at the 3.1 billion sites in it. And so, between anyone in this audience, we're more similar than we are different, but we do different in very little ways that do cause differences in traits. So one way is these genetic variants called single nucleotide polymorphisms. And basically, at a certain position in the DNA, one person might have a G and one person might have a C and vice versa. Or you might have 2 Gs and they might have 2 Cs and things like that. And at 1 in 1,000 places in our DNA, these positions happen and make us slightly different. We can have slightly larger changes called insertions and deletions or indels, where, I could have this extra CG in my DNA that just got there sort of by mistake or just once upon a time. And these can also cause differences and changes. And then there can also be these sort of larger, bigger changes in your DNA where chunks can get taken out or reversed or flipped around in all different ways. And all these different types of variations cause some of the differences that you see between us. So, these are changes at the DNA. They don't really do anything themselves. They basically enact their effect at different scales and different levels of biology. So DNA changes, DNA basically has the instructions for life. And so DNA basically gets converted into RNA, which is a message that gets made into protein. And the proteins are like the little machines in our cells that do all the different things that we need to live. But these DNA changes cause different levels of RNAs and proteins or different activities. So the proteins can function different ways or to have different behaviors based on your DNA. And these changes cause differences at higher levels, like organ function or like how fast your heart rate is, things like that. You know, these changes over time can result in different disease processes occurring. So maybe if you had higher cholesterol production in your liver, you know, you might have more plaque in your arteries, and that these changes then have an effect on your disease risk. So in a population, because of changes at the DNA, you'll see these changes in the end in disease risk. So genetics can explain only some of the inter-individual differences in the traits you're likely to have, like whether you're likely to be taller or shorter, and the diseases you're likely at highest risk for. And so the changes I showed, these SNPs can have influences on your disease risk. And we can measure approximately how much variation between each of the individuals in a population is maybe because of differences at the genetic level. And we call this quantity heritability. So how much variation at the DNA level is present at the sort of changes between people, the phenotype level, as we call it. And so, you know, your height, about half of the variability in height in a population is because of genetic variance. About 30% of the variation in your body mass is regulated by genetics, but about 50% of the risk of developing a heart attack, coronary artery disease is caused by genetics. So, you know, genetics is important, but it's not the only thing that happens. There's lots more that happens that's important. And so we can identify which exactly DNA changes these genetic variants are associated with diseases in research cohorts that have health records and genetic data. So if we have health records, we can take all the people in a study and say who's gotten a heart attack and who's healthy. So who's a case of a heart attack and who's a healthy control. And then we can measure a specific variant and say, like, is this variant associated with having a heart attack? And is it, you know, maybe that variant then causes heart attacks. And so using genotyping technologies like sequencing or microarrays and things like that, we can measure whether the variants are more in people with heart attack versus healthy controls. And in this example, you know, here's a variant that's in 58% of the heart attack cases, but only 16% of the controls. So this variant is associated with heart attack risk. And we call these studies genetic association studies, but when we do them for all the genetic variants we can measure in a study in a person, these would be called a genome-wide association study. And so by repeating this for lots of different genetic variants, we can generate these things called Manhattan plots. And so on the bottom axis, the x-axis is the position in your DNA where the genetic variant occurs. And on the y-axis is the strength of association. So how likely the change in that DNA is to be associated with heart disease, basically. And so we can also summarize these in sort of a written way by saying the variant ID, this is where the variant is in the genome. And then the alleles are basically like which DNA change, you know, changing from an A to a G is associated with the third number, which is a 1.21 odds increase, which is very small actually, but a 1.21 odds increase in developing the heart attack versus if you didn't have 2 copies of the G allele. So that's how we can measure which variants are associated with disease risk, but then we can go sort of use that information and summarize it to get an estimate of disease risk, because that's about the variants, but now we can, for a single person, basically get a measure of their genetic predisposition to almost any trait or disease that has these GWAS, these genetic association data. And so we call these predictions made with genetics polygenic scores or polygenic risk scores, because they predict your risk of a disease, or they predict some trait that using genetics. And the poly comes from that there's just lots of different variants that we include in these scores. So they can include maybe hundreds to millions of different variants that are associated with disease. And the genic part comes because we're using genes and variants, so the genic variation. So basically, we can get an estimate of the person's polygenic score by taking the genetics, so how many copies of each of the bad alleles they have at all the different positions in their genome. And then if they had 2 copies of all the bad alleles, so that's the maximum you could have, the maximum polygenic score, so the worst you could have is a score of 2.62. So in this case, I've made a little emoji person, and they have, 1 copy of some of the risk alleles and 2 copies of some of the other alleles that cause the genetic variants that cause heart disease. And so this person, when you sort of sum up their data according to that formula and the numbers in the first table, has a polygenic score of 2.04, which is pretty high compared to the maximum you could get is 2.62. But this other person, maybe she's a female scientist, so it could be Samantha or Amy, they're more likely than maybe me, the person in the tuxedo, and they don't have many copies of the risk alleles. So when you calculate their polygenic score, it's much lower. It's only 0.32, and that's lower than in the population. So this is basically how we generate the polygenic score, but how do we interpret what it means? Well, we know that in a population of people, and so here, if we took like a research cohort that had genetic data and health records associated with them, we can measure everyone in that cohort's polygenic score. And most people would have this mid or average polygenic score, but there would be people who are lucky, they're at the lowest end of the distribution, and they would have a lower than average polygenic score, and some people will have a higher than average polygenic score. And it's important to know like how these differences play out and whether these actually cause or constitute meaningful risk, differences in risk. And so we can measure using the health records, so information about the polygenic score, and then whether these people develop disease, to measure how different categories of genetic risk corresponds to future risk of heart disease. And so in a study from the lab I work in Cambridge, we showed that people who have the highest genetic risks for coronary heart disease have heart disease earlier and at a higher frequency than people who have lowered brackets of genetic risk. And so on the x-axis of the graph is the age at which we're studying the people, and on the y-axis is how many people at that age have had a heart attack or coronary heart disease. And the colors are the buckets of genetic risk. So you can see that the people who have the highest genetic risk have higher incidence or heart disease events over time. So I think people might wonder, researchers often wonder, which diseases and traits have polygenic scores, so which diseases can be predicted, but how well do they work? I showed you a pretty cool example where the polygenic scores work really well and strongly predict future heart disease risk. And so they want to know how well they work and who developed them, and what are the data and algorithms and people used and research cohorts used to make these polygenic scores. And so this data didn't exist, but part of my research was basically to answer these questions and develop a resource where people could look at these data, mainly researchers and clinicians who might want to use these in clinical practice. And so this database is the Polygenic Score Catalog, and it's the first and largest database of these polygenic scores developed all around the world from lots of different researchers. And we present them in the database so that people can use them in their own research data and study how they were developed and how well they work. And so anyone can look at the website, it's pgscatalog.org. And in the catalog now, we have over 2,000 different polygenic scores, and these predict over 500 different traits. So, and it's already being used to make lots of different research findings, and also some of which are being looked at for clinical translation. So in the next couple of minutes, I'll talk a little bit about the polygenic score research that's happening in Cambridge. So I told you on the last slide about how we curate and catalog all the information about polygenic scores so that people can get a really good understanding of the current state of the art in prediction. But in the next slides, I'll talk a little bit about why polygenic scores might be clinically useful and how they might be able to improve risk prediction. So Sam also talked about risk prediction, about how you can, doctors use information to basically make treatment decisions. And yes, doctors use risk prediction to make decisions already. So, at around age 40, you might get identified or invited into your GP for a health check. And, they'll use common predictors of disease risk, like your age, sex, your body size, lifestyle measurements about smoking, alcohol, your prior history of disease or medications you take, or also your family history, to basically make assessments about what diseases you might be most likely to develop and how we might be able to mitigate that. And, when we're making predictions about specific diseases, say cardiovascular diseases like heart attack and stroke, we measure different or slightly more specific risk factors like your blood pressure or your cholesterol measurements to make more informed disease-specific risk predictions. And so if you take this example, we can integrate all these information to measure your risk. And, there will be a lucky amount of people who don't have a meaningful amount of risk for cardiovascular disease in the next 10 years. But these people would usually be recommended, to continue their lifestyle, but then be reassessed in a couple of years' time to make sure that no risk changes have occurred. But these risk predictions can also identify the unlucky few among us who are at highest risk to develop heart attack over the next 10 years or something like that. And the doctors might encourage you then to make lifestyle modifications that reduce risks. So, exercise, smoking, reducing smoking, changing your diet, reducing alcohol intake, or also recommending you different medications that might reduce your risk. So if you have over a 10% risk of heart disease in the next 10 years, you might be prescribed a risk reducing medication like statins. But this information we know that genetics involve or has some effect on your disease risk, but this information currently doesn't involve genetics. And your DNA has this genetic risk information in it, and it stays the same from birth. So we think that maybe these polygenic scores might be useful to inform your risk of heart disease before any of these negative changes like high cholesterol or high blood pressure occurred. So, because your DNA stays the same from birth, we could calculate your polygenic score from birth for multiple disease and use this information over the lifetime to maybe prevent different diseases before risk factors occur, to make sure that people who are at the highest risk from heart attack in the future might always keep their risk factors like blood pressure and cholesterol low. And so polygenic scores could have this risk information before you present with sort of these high risk biomarkers in the future. And so this is just one scenario that polygenic scores could inform maybe risk prediction before or while you're still young, but they might also be used to alter screening regimens. So in cancer, we're not trying to prevent disease. Usually you're trying to identify early stage disease so that you can treat it before it becomes unmanageable. And so this is why people get mammograms or have bowel cancer screening. And so one might think that polygenic scores, because they're risk information that's available earlier, we could use this information to say, like, maybe we want to look at the people who are at highest risk for breast cancer or bowel cancer and say, like, let's screen them for early stage disease earlier in their life or screen them more often. And in this way, polygenic scores can inform the decisions about when we start screening. We can also use it to refine risk estimates using other risk factors like blood pressure and cholesterol or other rare genetic markers that are already used. And people might be familiar with BRCA mutations in breast cancer or prostate cancer as well. And so, we might want to refine these risk estimates, and the polygenic scores add a little more granularity to those predictions and can improve those for carrier screening. And finally, polygenic scores might be also useful for targeting those treatments to those who are most likely to benefit. So there is some research out there that suggests that people at the highest level of genetic risk for heart disease are most likely to benefit from statins versus people who are at a very low risk but have high risk factors and so on and so forth. So these are just some of the ways that, this risk information could be used to suggest who's likely to benefit from treatment or who should we screen and make sure that we try and prevent diseases in. So outside of the clinical aspect of polygenic scores, we can also use them to sort of dive deeper into the biology or the molecular underpinnings of diseases. And so we can use these cohorts and data to identify proteins that cause disease. And so it's basically getting at this, we're trying to go closer to the molecular, the cellular level to identify which genes are causing disease. And so we use polygenic scores in this way we can try to use them to identify these molecular drivers by first measuring your polygenic risk of a certain disease, but then measuring different levels of RNA and protein levels in a population in a research cohort where all this data has been collected. And then we can find the proteins that might be associated with disease risk by looking at proteins that are highest in people with high disease risk and lower disease risk, and then look in their health records to say whether these proteins are actually mediating or causing this disease in the future, basically estimating the mediation. And so we've been able to do this. A postdoc in the department, Scott Ritchie, led this study that was published last year, where we could show that, by studying in people who don't actually have any of the disease yet, healthy blood donors of the interval cohort that Amy measured. In this cohort, we had genotype and polygenic scores measured and lots of different protein levels for 3,500 different proteins in hospital records. We could basically find these proteins that do mediate disease risk, some of them which were already known in the case of chronic kidney disease. There's these two proteins that we identified as causal mediators of kidney disease risk. But these data could also be used to make new discoveries and also make discoveries that have therapeutic implications. So here, when we look at proteins that mediate type 2 diabetes risk, you could find this protein FBP1 that causes increased type 2 diabetes risk. And indeed, this is already a known target for anti-diabetic drugs, these gluconeogenesis inhibitors. And so, these tools allow us to find the proteins that we can drug and change disease risk with these drugs. And this was already known, but we could also then find other novel proteins like IGF BP2, which is a mouthful, but it stands for insulin-like growth factor binding protein 2. And this is a protein that, although we didn't know it was associated with type 2 diabetes risk before, but it is a protein that has the properties that make it a druggable target, that maybe we could develop drugs that could be useful for treating diabetes. So this would be an interesting finding from our study. So yeah, in summary, I told you a little bit about how genetics explains some of the inter-individual differences in traits and disease risks, and how we can measure and estimate these using polygenic scores. And I talked a little bit about, polygenic scores, how they could be useful in healthcare settings for personalizing disease prevention and screening regimes, and identifying the molecular changes that cause disease. And there's a lot of people involved. It's a team effort with many at Cambridge and abroad. And so these are just some of the people I work with, some of the nice people. But yeah, thanks for listening, and I'm happy to take questions.
That was lovely, Sam. Thank you. So we have about 10 minutes left. We have had some questions already in the chat, so I'll probably start with one of those. But if people have questions they haven't already put in the chat, I suggest they start, sorry, not in the chat, into the Q&A. I suggest they start putting them in so we can get to those ones too. Why don't we, in fact, I think I will start with the one right at the bottom that I just wrote a long answer to, but Sam probably also has some interesting things, where Terry says, I have heart disease, but it didn't develop significantly until I was 86. So depending on when my genes were measured, would I be high or low risk?
Yeah, that's a great question. So I think that, your disease risk does change over time, but there is some findings that say that the genetic risk is, if you have high genetic risk, you're more likely to develop a heart, for heart disease, let's say, you're more likely to develop a heart attack earlier. But some things like over time, genetics becomes less useful because, as I said, you have risk factors like high blood pressure and high cholesterol. So once we know that your risk factors for heart disease are higher, the genetics becomes less useful because we already know you're exhibiting early stages of risk for heart disease. So the genetic data, it always would say whether you were high or low risk from birth, but whether that information is useful changes over the lifetime, where it's most useful before we know anything about, your blood pressure or your cholesterol.
The other thing I said to Terry is I tend to try and think of the genetic risk as just a weird mathematical way of expressing family history. Like it's just a number that says, you know, if instead of saying, oh, I'm very high risk, you just mentally think, oh, my parents died of this, or all my grandparents died of this. You're probably thinking of a similar level of, well, it might not affect me. I'm a little worried it might. Whereas if all your grandparents died of something completely different in a different system of the body, you probably, that's more like being lower risk. I hope. We have another very interesting question, which I would love to know the answer to, and I imagine lots of people would, which is how will COVID affect CVD rates in the future? Of which the very short answer is we don't really know enough to know long term, but what we know short term, it doesn't look great. So either Sam or Sam want to expand on that anymore? Or...
So the earlier findings seem to suggest that there are some sort of longish tails to these things. So it can have quite a dragged on effect. So you might have heard of long COVID, the idea of long COVID. So we need to study more and see how long that lasts and etc. And how does it change by different subgroups of the population, like by age or ethnicities or so on and so forth. Or their histories and stuff like that. And yeah, and also there might be, oh, sorry, I just saw a chat that is addressed to me. Okay, so yeah, and also it also depends on, there might be some backlog from the government responses and so on during the pandemic. Maybe there are people with various issues that are not properly treated and so on. So we'll have to see what happens, and it'll be an exciting thing to study and hopefully, yeah, mitigate it as much as possible.
Yeah, I think the only thing I'd add to that is we are seeing some evidence of higher rates of CVD, even in the people who don't have long COVID. So it doesn't have to, you don't have to feel like it's continuing to affect you for it necessarily to possibly be raising population level rates of heart events. And obviously, even with the very, very long tails, part of the problem is if we keep having COVID infections, do we get the same effect again, or does it get worse over time? And we don't know, because we don't really have enough populations of people where we know they've had multiple bouts of COVID and when they've had them yet. So maybe, hopefully, something we can answer better in future. Okay, so we've got another question, which is, how long did this model take to create? And if it was created using a different target other than CVD, would it be flexible enough to reduce the time to produce another model? And I'm not sure, from the question, whether they mean the like polygenic risk score models or the national COVID model. But do either of you want to comment on, you know, how long would it take you to adapt your model to work for a different outcome?
For sure. I think, you know, when it comes to polygenic scores or risk predictions made using genetics, these polygenic these data sets that we use are usually, they take a while to create, but creating the model is usually a matter of like weeks to months, where we take the knowledge about the genetic associations and then we combine them with a computational algorithm to basically get which variants we should use to make these predictions. And then we evaluate them in new cohorts, new people, to make sure that these models work correctly. You know, we have to develop a new model usually for every disease, because the variants that cause a disease like heart attack won't be the same variants that cause a different disease like a specific cancer. So each model has to be created for a specific disease or a specific target in mind. But the methods we use to generate the model, these machine learning or statistical techniques, are the same across all diseases. So it's more a matter of making careful analyses to make sure that these models predict what you want them to predict and that they work as well as they should. Sam, maybe you want to add to that as well.
Yes, so I guess, okay, so how long does it take to change the outcome? Yeah, so it's mainly in defining the risk predictors, for example, that are specific to your outcome of interest, because they can be quite different. But it does obviously save quite a lot of overhead. For example, you might have built pipelines for cleaning data and so on. And that actually takes a lot of time usually. So having all these things in place, it actually does save quite a lot of time for future projects. To be honest.
I mean, I'd say as a general rule of thumb, the thing that takes the longest is finding the data and making sure the data is, you know, for example, if you have a rare outcome, it's much harder to find data sets large enough to contain it. We're lucky in that CVD is quite common. And so, you know, we only need 50,000, 500,000 people, and we've probably got enough to have a good study. But if you've got something that's only affecting 1 in a million people, you'd need far, far larger data sets often in order to be able to make a reasonable study on them. And then secondly, deciding what model it is you want to run takes longer than the computer takes to actually fit the model. So yeah, the next question was, can sleep affect the risk of developing CVD?
I've not studied it, but if it affects inflammation and stuff like that, it could potentially, I could potentially see a biological.
Might not be causal though. It could be, because it could be early impact of CVD making sleep difficult.
Yeah, potentially.
Yeah.
I don't know. Like I said, I've not studied it.
Yeah.
But also, like, are you not sleeping because of stress, chronic stress, anxiety, panic attacks, like I have? And maybe
the cheerful side, they might not be sleeping because they're out all night partying, taking lots of drugs and binging on alcohol. It's still better to have a heart disease, but at least they're having a good time. Okay, and then Cheryl asks, obviously you have robust data from those with medical problems which are recorded in detail. How do you ensure that your data is not skewed by this and that healthy controls are included in sufficient numbers?
I think that's a really great question, Cheryl, and I guess that's something we didn't talk about in our presentation, but you know, the types of data we actually collect or these studies they vary in different ways. So in my presentation, I talked about cases and controls where maybe we have a lot of people that we know already had a heart attack and we find some people who didn't. And this is sort of like a case control study. But a lot of the studies we work on are population-wide studies, or we go out into the community and recruit a representative sample of people that we don't know whether these people have disease, had disease, or what diseases they're going to develop. We just know that, you know, these are all the people who live in a specific area and we want to study them as a representative sample. And so this is really important, and it's what a lot of health data research focuses on, is trying to get these representative samples of people across age, sex, ethnicity, across the life course, to make sure that our studies and our findings can be most generalizable to everyone in the population, not just people who are you know, sick or people who are too healthy at the other end.
Yeah, I mean, UK Biobank has exactly the opposite problem, right? It's too healthy. It's representative that there are too few people with disease compared to the average population rather than too many. And so the skew has gone the other way. There's also a question that Hilary put in the chat, which was, what is DNA, what does it look at, and how do we look at it? Is everyone too far down in the pipeline of data to know the answer to that?
The DNA?
Yeah.
So DNA, it looks at, you know, you can't say, well, you can do sort of a home experiment where you swab your cheek and put it in some salt and get little strands of DNA. But the DNA is, yeah, bananas too. But, you know, DNA is basically just chemical letters that are made up of 4 different, it's only 4 letters, A, C, G, and T, and is 3.1 billion it's like a word that's 3.1 billion letters long. And so basically, you can't see it, but we know what it should, or what it looks like in the average person and what it looks like in specific diseases. So how we look at that is basically you get someone's blood or a cheek swab, and we extract the DNA from their cells, and then we basically put it on either a microchip or a DNA sequencer, which basically allows us to read the letters and see at which positions of the 3.1 billion characters in the word are different from what we would expect or what the average person has. So we basically turn these chemicals, these different chemicals at each position into just a string of, or just a typed list of A, C, G's, and T's on a computer that we study to look for which ones are associated with disease.
There's a little magic box. It's about this big, and you put your DNA on it, and it does magic. From my perspective, as a statistician who just gets the numbers out the other end. We have hit 7 o'clock, so we do need to wrap up. Thank you so much to everyone who came and asked questions and listened to us. You can always try and get in touch with the department via the Twitter or by emailing into us. Just give me a second. I will put the CEU Twitter handle, which I can't remember off the top of my head, unless someone else can remember it. Nope, can't find it. Yeah, we really appreciate everyone coming and listening to us, and just hearing the questions and hearing you talk about us. And just wanted to say thank you so much to Sam and Sam for joining me.
Great. Thank you everyone for joining, and it was great sharing our research with you.